The molecule in the lower left of the slide is called tetrodotoxin, or TTX for short. It is a potent neurotoxin found in multiple animal species, such as blue-ringed octopuses, pufferfish, frogs, and my favorite, newts. TTX blocks channels in nerve cells that sodium uses to enter and leave nerves, leading to muscular paralysis and the eventual asphyxiation of the organism that ingested TTX. What a way to go out. My favorite TTX-possessing organism is the rough-skinned newt, properly known as Pterica granulosa, as seen in the picture on the top left. Newts are salamanders, and this specific newt gets its name from the little TTX glands present on its dorsal surface. Recently, it's been discovered that TTX in this species may, in part, be produced by the newt's natural microbiota, a fancy term for bacteria, fungus, and whatever other microorganisms are naturally present on or within the newt. TTX has also been found in internal organs such as the ovaries. TTX needs to be transported from the skin to the ovaries through the bloodstream. However, blood plasma is mostly water, and TTX does not dissolve well in water. So how does it get there? It teleports. Wait, no, that's a little too much physics for me. It's much more likely that there exists some sort of molecule that transports TTX through the bloodstream, since on its own, TTX isn't getting far. This brings us to why I'm here. TTX binding protein is a protein that binds to TTX that has been identified in pufferfish, but is yet to be found in newts. It's currently unknown how TTX is transported within the bodies of the rough-skinned newt, but we hypothesize that it is by a TTX binding protein. Our experiment is to isolate and identify TTX binding protein in the blood serum of the rough-skinned newt. To do this, we are using a co-immunoprecipitation assay with a novel twist. An easy way to describe co-IP, as it's known for short, is it's kind of like fishing, as illustrated in the top right figure. A primary antibody, or bait, is used to catch a protein, the fish. Then a secondary antibody is used to reel in the primary antibody protein complex. The protein is then identified using known methods. In my case, however, we don't have the bait to catch TTX binding protein on its own, as it hasn't been discovered yet, and bait is not available for undiscovered proteins. We need to flag it with something we do have the bait for, TTX. After binding the protein to TTX, we can then do co-IP using TTX antibodies as the primary antibodies, which bind to the binding protein TTX complex, like in the bottom right figure. By doing this, we hope to isolate and identify TTX binding protein and understand how TTX is transported within the body of the rough-skinned newt. Thank you.
Hi, everyone. I'm Misbah Chaudhry, and the title of my project is Exploring the Biocontrol Potential of Streptomyces species against Pythuym violae, which is a dominant carrot pathogen.
Okay, on the count of three, are you ready to start?
Yes, sir.
1, 2, 3.
The United States is among the largest carrot-producing countries in the world. California ranks first with the production of over 85% of all the carrots grown here in the US, with Kern County accounting for 60% of that production, which makes our agricultural sector significant around the nation. However, with such a great production comes a downfall for our farmers and growers in the form of diseases that carrots can be affected with. Because these diseases not only affect the overall yield of the carrots, but they also reduce their market value as these diseased carrots get rejected in the market. Now, out of all the common diseases, my project is focusing on a disease called cavity spot, which is of a great concern here in the Kern County because it affects over 50% of the overall acreage. This disease is caused by a fungal-like microorganism called Pythuym violae, which affects or infects the carrot roots as can be seen in the image on top left. And it causes for these sunken brown to black lesions to form on the carrots. This disease is difficult to manage and is currently being controlled with chemical fungicides. However, these fungicides are unreliable because of the resistance from the pathogen, and also generally they're just harmful for our environment. So the overall goal of my project is to rather look for a more natural method in a form of a natural organism that can be used to control these diseases or disease. So for that, I have been exploring species of a bacterium called Streptomyces, which is also a microorganism found in the soil and is known for its production of these natural products that actually inhibit the growth of these pathogens and show antifungal activity. And as some promising results show, and as you can see in the image on the bottom right, some of these species do actually inhibit the growth of the pathogen that causes the cavity spot disease, which means we can use them as biological agents controlling this disease and therefore resulting in good quality carrots that you and I can enjoy. So with my study, I will be putting forward a selection of these Streptomyces species to initiate the formulation of a commercial biofungicide that can be used as a safe, natural alternative to the chemicals that are currently being used in our fields, in carrot fields, to not only help our agricultural sector, but to also provide for a more safe environment for everyone. Thank you.
Yes, my name is Amy Fetters, and today I'll be presenting on riverine litter and how bridges impact the distribution of anthropogenic litter in a freshwater river.
Thank you very much. On the count of three, are you ready to go?
Yes.
Okay, 1, 2, 3.
All right, so we have all heard about the plastic straw in the sea turtle's nose, or the massive patches of garbage floating in our oceans. As humans, it's natural that we produce waste, but what's not natural is when a majority of our trash ends up in the environment. The global production of plastics alone is approaching 400 million tons each year, with only a small percent being recycled. Scientific research all over the world has focused on trying to figure out how anthropogenic litter, or man-made trash, is affecting our ecosystems. Most research on litter occurs in marine habitats, with less attention directed towards freshwater. The amount of litter in freshwater rivers is increasing as human populations rise, and rivers often serve as a major source of debris in our oceans, so it's important to understand how much litter is in freshwater and where exactly it is distributed. In addition to rivers, humans have also altered in addition to pollution, humans have also altered rivers by constructing man-made structures, such as bridges. When structures are present in the riverbed, materials that are flowing downstream have the opportunity to accumulate on these obstructions that disrupt the normal flow of water, resulting in the formation of debris dams, such as the picture in the lower right corner of my slide. I have found that these debris dams, which are typically made of natural materials like sticks and leaves, are now in part made of anthropogenic litter. My slide here is summarizing the goal of this study, which is to determine if the presence of a bridge with closely spaced columns in the riverbed impacts how much anthropogenic litter is being transported downstream due to the presence of debris dams underneath the bridge. So to collect my data, I collected anthropogenic litter upstream of the bridge, downstream of the bridge, and underneath the bridge, where all of our debris dams were located. From my preliminary data, I found that the amount of litter upstream versus downstream showed no significant difference from this first sampling event. However, debris dams contain the highest density of anthropogenic litter items and also the highest abundance of plastic items specifically compared to all other locations sampled. These results support that debris dams do capture a high abundance of litter that is flowing in the river, potentially altering the amount that is being transported downstream. Also, debris dams capture a high percentage of plastic items, which are particularly harmful for the environment. Overall, from this study, I was able to gain insight on where litter is distributed in rivers, and this information can be useful for trash monitoring and litter removal programs that are working to fight and eliminate plastic pollution in our environment. Thank you for listening.
Long time ago, over 4 billion years ago, that is, the moon and Earth were constantly being bombarded by giant asteroids and failed planets, basically the leftover planet building material of our solar system at the time. And some of these impacts we think were so big that they were actually able to excavate and eject lunar mantle material out into space. And we think that over some time, some of that material eventually fell into Earth's orbit and crashed down here in the form of a lunar meteorite. And so what you're seeing in the upper left corner, the picture there, is an actual piece of the moon. And that piece of the moon traveled 240,000 miles through space and eventually crashed down here in the desert plains of northwestern Africa. And one thing to know about lunar meteorites is that they're extremely rare. There's only about 150 of these known in existence. So you can imagine how special an opportunity we have to research something like this. And so over the course of the next 2 years, we'll be conducting a series of lab tests and analysis, like electron microprobe analysis and laser ablation mass spectrometry. And what we hope to gain through those are basically a history of the crystallization of the minerals that make up this sample, and also a geochemical composition of the lunar mantle that it was derived from. And so when we have that information, we'll be able to answer a myriad of questions like, when was it actually formed? When did it crystallize? How deep in the mantle was it? And we'll even get an idea of the internal structure and stratification of the lunar interior, which we still have a lot of questions about that. And so what we really want to do with this research is build on all of the knowledge that we currently have from the Apollo missions and the samples returned there. But we also think that we'll be able to reaffirm or even challenge some current suspicions that we have about the lunar interior. But we also might make a potentially completely new discovery. One of the interesting things about lunar meteorites is that they all have a high probability of coming from a location on the moon that no lunar lander or astronaut has ever been to. And so I'm really looking forward to bringing everything that this sample has to the scientific community. And hopefully in doing so, increasing and furthering our knowledge and understanding in the field of lunar science. Thank you.
How many memorable interactions from childhood can you recall with a parent? Were they loving, heart-filled, or perhaps even harmful? And how has this shaped your identity today? These are arguably some of our most formative experiences. They undoubtedly shape our identities throughout our lifespan. Full disclosure, I am a mother in psychology. I imagine that I knew at least the basics of communicating positively with my sons. However, as a therapist trainee, I started to notice something really intriguing. Families often report really different narratives surrounding their values and experiences regarding mental health. I started to wonder whether the message that I'm trying to send my children is actually the message that they're receiving. My research utilizes a qualitative dyadic approach to understand what contributes to this communication about mental health. By interviewing pairs of parents and their adult children in private interviews, we can then examine what is contributing to these really vastly different perceptions. Much of the previous literature evaluates this phenomena by talking individually to either the offspring or the parents. However, individual experiences and thus our relationships are colored by a variety of different factors, our emotions, culture, even the historical context of these interactions. So utilizing only one informant to understand this is a little like trying to understand a piece of literature if you've only read half the book. Current research suggests that this matters. Parent-child discrepancies between perspectives, For example, how they see severity and symptoms can actually negatively affect even treatment outcomes in psychotherapy and our motivation to seek help. So from both literature and our clinical experience, we expect that we will find discrepancies. We expect that mental health is often defined negatively. I actually wonder if anyone here at hearing that even knew that there was another way to see it. I imagine some messages spoke louder than others, in that no communication about mental health was a communication about mental health. Beginning to understand this is crucial for anyone who interacts with children, because we have the capacity to build on their resilience. However, the most powerful utility of this data isn't with the children, but rather with the parents. This data can help us to educate and empower parents to support their children. They are the ultimate stakeholders after all. And this isn't about achieving any ultimate truth in our participants. It's about understanding the process rather than the content. Each perspective that we will hear, whether it comes from the child or parent, is just as real as its pair. It is in beginning to understand each of these realities that we can then integrate these experiences to contribute to both their individual mental health and a cohesive functioning family system. Thank you.
So every year, billions of pounds of produce and decorative plants are damaged by weather, pests, and disease. Leafy gall syndrome is a plant disease caused by the bacterium Rhodococcus fascians. It affects many different types of plants and therefore causes great economic losses each year for farmers and nurseries. Recently, R. fascians was identified as a culprit of a new disease called pistachio bushy top syndrome. Since 2011, over 2 million infected trees have been removed in California, New Mexico, and Arizona. Decorative plants are also greatly affected, with a single wholesale nursery reporting yearly losses of $1 million due to R. fascians alone. R. fascians causes disease by producing plant hormones, the exact same ones that are also produced by plants themselves, despite these organisms being unrelated. Plant hormones control a wide variety of functions during growth and development and must therefore be tightly regulated. The additional hormones produced by the bacteria during infection are enough to upset the hormonal balance of the plant and cause deformations such as tumor-like growths seen up close in the image on the right, resulting in a stunted, bushy plant appearance. It was recently discovered that R. fascians produces unique hormones that are not produced by plants. These unique hormones behave in a similar manner as the traditional hormones, but the plant has more difficulty breaking these down, possibly making it difficult for the plant to defend itself against the bacterium. My research aims to determine if these unique plant hormones play an important role in the bacteria's infection process. To test this, I'll take away the bacteria's ability to produce these unique hormones and compare the symptoms caused by the defective bacteria to those caused by the normally functioning bacteria. If the symptoms are less severe without the unique hormones, like we predict, then we can conclude that they're important for infection. These results will further unravel how R. fascians is able to infect plants and hopefully one day aid in the production of prevention and treatment strategies, which will therefore reduce economic losses due to infection and hopefully conserve precious environmental resources such as water. Thank you.
Revolution is not a dinner party, and America is a paper tiger. And if this statement seems like a non sequitur, then congratulations, you're probably not a communist. However, if this statement does ring a couple of bells, then you may need to brush up on your little red book, comrade. This little red book is also known as Quotations from Chairman Mao Zedong, who you see on the left-hand side. He was the leader of Communist China from 1949 to 1976. And this little red book espouses all of Mao's most revolutionary thoughts, also known as Maoism. Maoism is a communist ideology, so you may assume that it has had minimal impact on the United States. However, this assumption dismisses the great influence that Maoism has actually had on certain populations of Americans, specifically those fighting for their rights in the 1960s and 1970s. My research questions how Chicanos, Mexican Americans fighting for equality during this time period, looked to the principles of Maoism to bolster their own movement and their own fight against internal American imperialism. So, following the American annexation of the Southwest, Chicanos were treated as colonized subjects and greatly discriminated against in their own historical homeland. My research has found that Chicanos saw in Maoism a pragmatic ideology that allowed them to view American imperialism in surmountable terms as a paper tiger. This then led to the formation of the Chicano national question, which advocated for the creation of a sovereign Chicano nation inside the territorial United States. My research has also found that Chicanos recognized from Maoism that their movement or their revolution was not a dinner party, it was an insurrection, which led to the formation of militant Chicano organizations such as the Brown Berets, who you see on the right hand underneath Mao, to protect this movement or revolution. However, there exists little scholarly discussion that combines the influence of Maoism and the Chicano movement together. Although there does exist multiple sources such as letters, articles, declarations that do so that had not yet been engaged with that I utilized in my research. Due to this lack of prior work, my research pulls from the methodologies of historians that examined Maoism, but its impact on the African-American civil rights movement. And then I applied their historical framework onto the Chicano movement. Lastly, my research endeavored to look at these sources that I mentioned using only the Chicano perspective to ensure that Chicanos were given their own unique historical space during this time period in American history, and therefore respecting and honoring the Chicano people and the Chicano movement in this period of history. Thank you.
Potatoes are the leading vegetable crop in the United States. In 2019, it brought in a revenue of $3.94 billion. So as you can see, potatoes are economically important, but there is some jeopardy in their market value. And this is because they can get a disease called common scab disease. As you can see on the slide, the potato on the left is a healthy potato, and the potato on the right has acquired the disease common scab. What happens to the potato is it develops these scabby lesions on the surface, and this decreases their market value because it becomes undesirable to the consumer. The pathogen that's responsible for this infection is the bacteria called Streptomyces scabies. What this pathogen does is it produces a toxin that doesn't allow for the potato plant to grow appropriately. So when this potato plant is trying to grow and stretch, it ruptures, and this then allows the pathogen to enter and cause the disease. But what's intriguing about this infection pathway are the subunits that are found in the soil that are telling the pathogen it's time to make this toxin. If you focus again on the slide at the bottom, you can see that these subunits are coming from living and dead plants. So how does the pathogen distinguish between the two, and can it even do it? I predict that the pathogen can identify between the two. And I think this because I have learned that the pathogen invests a lot of energy in order to produce this toxin, because it uses these really big enzyme molecules in order to assemble the toxin. So it's not going to want to waste this energy, the toxin, on a dead plant because it can't steal nutrition from it. So how I will investigate this is by infecting living and dead potato plants with the pathogen and checking for toxin production. If the pathogen can identify the living plant, I will further investigate how. Something living plants can do is excrete material from their roots, such as amino acids, or organic acids, or sugars. I can collect these root exudates and see if these are used with a pathogen to see if there's a living plant there. This research is important because understanding how a pathogen recognizes its host will help us to prevent the infection, because we can then prevent the interaction. This research is not only important for the potato industry, but also for other tuber crops, because this pathogen has also been recognized to cause disease to carrots, radishes, and even peanuts. Thank you.
Some single-celled organisms move around using tail-like appendages called flagella that allow them to swim in watery environments. Others can move along semi-solid surfaces using retractable arms called twitching, and others still can move along watery solid surfaces by making it slippery, allowing them to start sliding along. These are all different forms of motility, or ways to move and be motile. My research focuses on trying to figure out how a soil plant pathogen can get to its ideal environment, a plant host. Rhodococcus fascians, or R. fascians, is a bacterium that causes a leafy gall syndrome. As you can see in the photo with the two plants, the one on the right is nice and tall and healthy, while the one on the left is infected, it's short and it's stumpy with its leafy gall. Rhodococcus fascians can actually mess up a plant's hormone balance and make a plant tumor home for itself. This is bad for both ornamental plants because it makes them look bad and unmarketable, as well as reduce agricultural plant yields. A recent example of this is our fascians infecting pistachio trees, not allowing them to produce pistachios. This is a big economic burden for the growers who rely on selling pistachios for a living. Our fascians has long been described as non-motile because of the standard lab conditions it's been studied under and the obvious lack of a flagella. Remember, bacteria can move in different ways in different environments. Well, it was later found that growing on a lower concentration of agar or a more watery jello-like surface, R. fascians was actually motile. The photo on the right shows a normal R. fascians spreading compared to a mutant. Mutants are just ways of how I study the organism. What a mutant is essentially where I take a specific protein and either knock it out of the organism or make it more abundant in the organism. And these are really good tools to help me visualize what impact these proteins might play in its motility. This lets me get a better idea of what the bacteria needs in order to actually move. So why is knowing how a bacteria moves important at all? Remember, as mentioned earlier, this pathogen has the potential to cause severe economic damage to both ornamental plants and for people who sell plant products for a living, such as fruits and vegetables. So this basic research question of how does a bacterium move can help fill in that gap of how the bacterium gets to its host and hopefully potentially mitigate its spread. Thank you.
The year 2019 was the second warmest on record, second only to 2015. Global temperatures have increased by over 2 degrees Fahrenheit as climates change. This has increased the risk and severity of drought. We saw this in the historical California drought of 2012 to 2016 that lasted 376 weeks. Drought is ongoing for Southern California. How does extreme drought like this affect plant communities? Well, less water is not good for plants, but it may be worse for some plants. During the peak of the 2012 to 2016 drought, some plants of Southern California were dying at high levels that were up to 90%, while others nearby appeared to be unaffected. You can see this difference in the plants in the left image. Some are dead, but some are not. Why? Answering this question is important for understanding the impact of drought on plant communities and protecting vulnerable species. One explanation is that these plants have differences in their rooting depth. During a drought, soils dry out from lack of rain inputs and continued outputs of evaporation and water loss. It is logical to think that those with deeper roots would be less affected by drought because they can have access to deeper soils that have more water, shown in the top right figure. Conversely, shallow-rooted plants like the ones in the bottom right figure may not be able to withstand the extremely dry soils. This prediction aligns with what was observed during the 2012 to 2016 drought. The ones that experienced higher levels of mortality are thought to be the ones with limited rooting depth. However, data are limited on the rooting depth of plants. It's not easy to dig up the roots of large plants to see where they go. Other Methods can be used involving analyses using water chemistry to trace where roots are accessing the water. This method takes advantage of the differences among elemental chemistry. Isotopes are rare forms of elements that are stable and measurable. For my research, I'm going to take soil samples ranging from the surface to very deep. Then I will extract plant water and match them to the soil samples using these isotopes. By doing so, I should be able to find out where plants are getting their water from. From this data, I hope to find out the rooting depth of plants and compare this to the data on plant drought vulnerability. If successful, I can predict which plants will be the losers and winners in future droughts. Thank you.
Shift work exists in many industries. Firefighters, truck drivers, doctors, nurses are some of night workers. These essential workers are exposed to serious health problems because working at night. Why working at night make them exposed to this serious problem? Because they're working against a circadian rhythm. What is circadian rhythm? It's a biological clock which regulates the release of certain hormones in the body that control mood, body temperatures, and alertness, and other aspects of the body's daily cycle. Research studies show that night shift sleepiness increases risk of job-related injuries and accident. Not only that, the sleep deprivation disrupts the body's metabolism that leads to long-term health problems, as shown on the top left picture on the slide, such as high cholesterol, obesity, insulin resistance. That is pretty bad, right? However, let's take a look at the top right picture. Many studies also show that napping during shift work can boost productivity, alertness, and even improve heart health. And now that is interesting. Here is where our research comes in. My colleagues and I wanted to know from night shift workers' perspectives if napping actually enhances productivity and alertness. After we received the IRB permission for us to study on human subject, we use emails and media and social media to deploy the link of our 16 question survey that we had developed for night workers who are interested to participate. The graph on the bottom of the slide shows the results of our study. There were 130 night workers responded to our survey. As you can see, that more than half of the participants, reported the improved work performance after a nap. And only 2.3% of those who took nap reported of extremely sleepy during driving home. So we know that napping is good. However, we also found a lack of nap rooms and limited support from management, preventing those night workers to be able to take a nap. Perhaps more studies need to be done on these barriers. Thank you.
The winner of our today's presentation will get a very nice clock with the CSUB Grad Slam first place 2020. It works. I tested it earlier when I was taking the picture. So I believe our judges have the scores, and I'm going to ask, earlier our judges took a vote on who will present in the first place. So I'm going to call on Mr. Jeremy Adams, who was chosen to give us the news of who is won the competition today. Adrian, are you able to spotlight Mr. Jeremy Adams, please?
So do I need to go down to the very bottom?
No, yes, yeah, go to the very bottom. And then look at the scores and let us know who got the highest score from the 4 judges.
Okay.
It's on column on the row 110, right?
Yeah, no, I just want to make sure I don't screw up as I do this.
Did you get your undergraduate degree from us too, or?
I did not. I got my Washington and lay back in Virginia, but I was a proud master's degree there.
Oh, wonderful. Thank you.
Okay. I'm pretty sure I'm seeing this correctly. I'll put on doing this on the fly.
No, I'm the second pair of eyes. Don't worry. I'll back you up. Okay.
Yes. Okay. So it looks to me, if I'm getting this correct, that your winner is Mrs. Mckinzie. Is that what you're seeing, Dr. Vega?
Yes, Hannah Mckinzie. And before we ask her to step to the front, let me say that this is just a testament to the presenters today, that there are ties, right? There are 3 of them that are 2 of them have the same score, and all of these are in the 4.0 and up. And what is also pretty amazing is that I asked the judges to use a decimal point to avoid the overlap. This is how good your presentations were, these have these high ties, and the difference between number 1 and number 2 is a matter of 0.02 or 3. So it was a close competition. We cannot emphasize enough, how good all of you. This is a win for all of you. So, we really give a big hand to you. So, Hannah, you're the person who scored the highest points today. Again, by a small margin. You had tough competition. We want to congratulate you. Is there anything you want to say or anyone you want to thank? So we'll give the floor to you.
I just want to say thank you, for this whole competition, and you guys were awesome. I was honestly sitting here thinking, they don't remember me. These guys are awesome. So I hope you guys realize that your guys' speeches, even though I was the only history one, I still got what you guys were putting down. I really liked it. So congratulations to you guys. You guys were great.